Welcome everybody. We have now this oral session about endophenotypes. You know the rules by now. You can have put your questions in the Q&A space. We will have a brief Q&A after each presentation, and then we will have a discussion at the end of the symposium. So I think we can start with the first presentation. Leanna Hernandez from University of California. Phenotypic and genetic association between childhood sleep, mental health, and brain structure. So Leanna, floor is yours.
Hi, my name is Leanna Hernandez. I am a postdoctoral fellow at UCLA, and today I'll be sharing our work examining phenotypic and genetic associations between childhood sleep, mental health, and brain structure. I have no financial disclosures or conflicts of interest to declare. Sleep is a biological process that's critical for optimal physical, cognitive, and emotional functioning. And it's well known that changes in sleep behavior occur across childhood and adolescence, including reduced sleep duration and later sleep onset and wake times. Childhood is also a developmental period characterized by changes in sleep physiology. These graphs show age-related changes in sleep spindles, which are short bursts of oscillatory brain activity that occur during sleep and are associated with sleep-dependent memory consolidation. As you can see, there's an increase in sleep spindle frequency during childhood, and this is also a period of peak spindle density. Importantly, however, sleep problems are surprisingly common in children, affecting 20 to 30% of the pre-adolescent population. And sleep disturbances are frequently comorbid with neurodevelopmental psychiatric disorders, being a core symptom of major depressive disorder, anxiety, and PTSD. Interest in children's sleep has seen a recent surge, in part driven by concerns over increases in children's screen time and late night light exposure, which is linked to both psychological stimulation and alertness, and may contribute to later bedtimes and decreased overall sleep time. Adult genetics research has shown that sleep behaviors are polygenic, with hundreds of genetic variants acting additively to influence sleep behavior, and genetic variants associated with sleep are enriched for genes expressed in subcortical and cortical brain tissue. Sleep-related genetic variants also show significant genetic correlations with adult psychiatric disorders, including depression, anxiety, and ADHD. And well-powered genomics research indicates a significant negative genetic correlation between adult sleep and brain surface area. However, parallel analyses of the shared genetic architecture between childhood sleep and brain structure have not been performed. The largest genome-wide association study of childhood sleep to date comes from the EAGLE Consortium, which found that SNP heritability for childhood sleep duration was 0.14 and a single genome-wide significant hit on chromosome 11. Notably, the same study found that the genetic correlation between childhood and adult sleep duration was not significant, suggesting perhaps some distinct genetic architecture between childhood and adult sleep duration. Only very recently have large-scale and population representative studies of childhood sleep emerged, with this recent study showing that longer sleep duration during childhood is associated with larger cortical surface area in frontal, temporal, and parietal cortex. However, to date, these large-scale imaging and genetic studies in children have focused specifically on sleep duration. So here we set out to investigate the genetic underpinnings of multiple childhood sleep behaviors by performing a comprehensive characterization of phenotypic and genetic relationships between childhood sleep, brain structure, and psychiatric symptoms, which brings me to our goals. So we had three aims. First, we examined how variability in multiple domains of childhood sleep is related to psychiatric symptoms and brain structure. Second, we assessed the extent to which sleep-related traits are heritable and genetically correlated with childhood mental health and brain size. And third, we tested whether polygenic risk scores derived from adult genome-wide association studies could predict childhood sleep behaviors in children 9 to 10 years old or at a follow-up time point 1 year later. So to address these questions, we use data from the Adolescent Brain and Cognitive Development Study, which is an ongoing longitudinal investigation being carried out in the United States. Participants are over 10,000 multi-ethnic youth who were recruited from schools within geographical areas that represent the socio-demographic diversity of the national population. So at baseline, children were 9 to 10 years old and visited a participating research site to complete a magnetic resonance imaging scan, assessing brain function and structure, cognitive tests, mental health questionnaires, and to provide biospecimens. 3 to 6 months later, participants were contacted by phone to complete a short interview about changes in mental health or substance abuse. At the 1-year follow-up, participants were 10 to 11 years old and again completed in-person behavioral assessments and provided biospecimens, followed by another mid-year phone interview. And the second wave of brain imaging data was collected at the 2-year follow-up visit when youth were 11 to 12 years old, in addition to cognitive and behavioral testing. And it's planned for this pattern to continue until the youth are between 19 and 20 years old. So at this time, all of the baseline data and half of the year 1 follow-up data has been released, and that's what I'll be presenting today. So to measure sleep problems, we use the Sleep Disturbance Scale for Children, which is a parent report questionnaire designed to identify sleep disturbances over the past 6 months in kids with or without clinically significant sleep disorders. And in contrast to previous neuroimaging and genetic studies, which have focused narrowly on sleep duration, here we examine the heritability of multiple common sleep-related disturbances, including insomnia, sleep-breathing disorders, disorders of arousal, sleep-wake transition disorders, disorders of excessive somnolence, and sleep hyperhidrosis. And for each of these subscales, higher scores indicates more frequent sleep problems. Good quality structural brain imaging data was obtained for 8,856 9 to 10-year-old children, including measures of whole brain and regional surface area, cortical thickness, and volume. The sample was randomly split into a discovery and replication cohort, and a series of linear mixed effects regression analyses were performed to test whether sleep predicted psychiatric symptoms and brain structure. For the genetics data, ancestry was determined by merging the ABCD genotype data with data from the 1,000 Genomes Project, and analyses were restricted to youth of European ancestry. A genetic relationship matrix was computed and thresholded at 0.05 to remove closely related individuals, and the GCTA toolbox was used to estimate SNP heritability and genetic correlations between traits. For our polygenic risk score analyses, the data were imputed to the top med panel. And after quality control, we retained roughly 3,500 participants at baseline and 1,700 who had follow-up behavioral data 1 year later. And polygenic risk scores were generated using PRSCS and trained using weights from the largest available genome-wide association studies. So first, we tested the relationship between each sleep disturbance subscale and behavioral measures of psychiatric symptoms and cognition. So to orient you, on the y-axis, we have p-values, and on the x-axis, we have each of the psychiatric and cognitive subscales examined, and the colors represent each of the 6 sleep disturbance subscales. So as you can see, across all sleep subscales, more frequent sleep disturbances were broadly associated with higher levels of psychiatric symptoms, with the most significant effects being found between insomnia and daytime sleepiness, with depressive and thought problems. Next, we examined the extent to which childhood sleep problems related to childhood brain structure. When examining the relationship between sleep and global measures of brain structure, we found that only insomnia showed reproducible associations with reduced surface area and volume. Turning then to regional measures of brain structure, here again, only insomnia showed reproducible associations indicated by the stars, with more frequent insomnia symptoms being related to reduced surface area in frontal, temporal, and parietal cortex, and smaller volume of cerebral white matter. No other sleep disturbance subscale showed replicable effects, suggesting that childhood insomnia is uniquely associated with brain size. Significant SNP heritability was observed for multiple traits, including insomnia, which is shown here with an estimated heritability of 0.15, as well as several of the externalizing related behavioral traits, which are shown in black, cognition in light blue, and brain surface area, cortical thickness, and volume, which are shown in dark blue. No other sleep disturbance subscales showed significant heritability. We then examined genetic correlations between traits that showed significant SNP heritability. So here, one of the highest genetic correlations that we found was a positive genetic correlation between childhood externalizing and ADHD. Focusing in on insomnia, we found positive genetic correlations with ADHD, externalizing, and total problems, indicating that the genetic etiology of childhood insomnia and these psychiatric traits is partially shared. Notably, and in contrast to the adult literature, we did not find a significant genetic correlation between childhood insomnia and brain size. Finally, we trained polygenic risk scores using a well-powered genome-wide association study of adult insomnia and looked to see whether genetic risk scores predicted childhood insomnia at either the 9 to 10-year-old baseline time point or 1 year later. We were surprised to see that there was no association between insomnia polygenic risk scores and childhood insomnia symptoms at either time point, despite the fact that the insomnia GWAS was well-powered and we had already observed significant heritability of childhood insomnia in this cohort. Further, binarizing into groups of youth who experience high, being in the top 25th percentile, and low insomnia symptoms, we again found that insomnia polygenic risk scores were not able to predict group membership. So then what does predict childhood insomnia? Given the robust genetic correlation we saw between childhood insomnia and ADHD, we generated polygenic risk scores for ADHD and found that they predicted both continuous insomnia symptoms at baseline and distinguished between youth experiencing high and low insomnia symptoms at baseline and the 1 year follow-up. To further tease apart the phenotypic association between insomnia and childhood brain size, specifically whether childhood insomnia drives small brain size or if brain size drives insomnia symptoms, we generated polygenic risk scores to predict the genetic component of brain surface area using the largest available GWAS from Enigma. So first we tested if polygenic risk scores for insomnia predicted cortical surface area at 9 to 10 years of age, finding no evidence that the genetic component of insomnia drives childhood brain surface area. We then tested the reverse relationship, finding that polygenic risk for brain surface area also did not predict childhood insomnia. So next, we wanted to see if the genetic overlap between childhood insomnia and ADHD could account for the association between childhood insomnia and surface area. And supporting this hypothesis, we found that ADHD polygenic risk significantly predicted both insomnia, as I showed on the previous slide, and smaller brain surface area in several of the brain regions where we had previously seen a phenotypic association with childhood insomnia. We also saw that the association between insomnia and brain surface area disappears when conditioning on ADHD. So altogether, these findings suggest that small brain size is not necessary for insomnia, and instead that the phenotypic association between childhood insomnia and brain size is driven by the pleiotropic genetic relationship between childhood insomnia and ADHD. So in sum, we find that childhood insomnia is heritable and genetically correlated with childhood externalizing behaviors, that insomnia symptoms are phenotypically associated with smaller brain size, and we found that polygenic risk scores for ADHD predicted not only externalizing behaviors at baseline and follow-up, but also insomnia symptoms at both time points and regional brain surface area, suggesting that childhood insomnia may lie along the dimensional axis of childhood ADHD. And finally, as insomnia polygenic risk scores trained with adult GWAS data did not predict childhood insomnia symptoms, there may be some distinct genetic mechanisms underlying childhood and adult sleep behavior. So if you would like to learn more about this work, a preprint was recently posted. And with that, I would like to thank my primary mentor on this project, Dr. Mike Gandel, my collaborators and funding, and especially the ABCD Consortium for providing such a great data set. Thank you.
Thank you, Leanna. Very nice, clear presentation. One question from Philip. Maybe, Leanna, you can see it.
Yeah, so it says insomnia has usually been found to have genetic associations with internalizing disorders. What do you make of the stronger pattern with externalizing disorders? So my thought there is that these kids are pretty young. They're just 9 to 10 years of age. And so there really isn't a great level of internalizing slash depressive sort of symptoms going on yet in these youth. And so I wonder if the pattern may change as they progress through adolescence and sort of move into puberty. And that's something that I'll be interested in looking at longitudinally.
Okay, I think that there are other questions, but maybe I may ask Leanna to answer to the questions in the Q&A so that everybody can read it and we don't go too much further.
Sure, thank you.
By the way, as a small comment, I think that it's very interesting that your insomnia in child is different from adulthood. It makes sense also from a clinical point that in childhood is more linked to ADHD and in adults more to anxiety and depression, and they have a very different genetic background. So very interesting piece of information. So for the next speaker, give the word to the co-chair, Orland Rajan.
Thank you. So we are moving on to a new endophenotype. This is creativity. And the speaker is Dr. Veera Rajagopal from Aarhus University in Denmark. He's going to present the large-scale genome-wide association study reveals first insights into the genetic architecture of creativity. So please, floor is yours.
Thank you. I thank the organizers for the opportunity for this presentation. My name is Veera Rajagopal. I'm a postdoctoral researcher in the Department of Biomedicine in Aarhus University. So today I'm going to talk about a project that I've been working on for the past 1 year in collaboration with many other scientists. So before I start my presentation, nothing to disclose. So this will be the structure of my presentation. So I will start with a background. Then I will tell you the study design and explain the results and the methods, and finally will summarize the findings. So first, background. So this is a GWAS of creativity. So what is creativity? So it is actually very difficult to precisely define creativity, but to put it in a simple term, it's the ability to create something different, something unique, which cannot be created using conventional thinking. So in other words, thinking outside the box. So how do you define creativity, or when you say someone is creative? So like individuals in any profession can be creative, but it is often individuals in some specific professions, such as musicians, writers, painters, sculptors, are often historically viewed as creative people. So how much as genetics has a role in creativity? So according to this study, which is based on twins and siblings, the heritability of creativity is as high as 70%. So why we are specifically interested in this phenotype is because it has a very interesting link with psychopathology. It has been known for centuries. There are many epidemiological studies that actually show that individuals with psychiatric disorders, particularly schizophrenia and bipolar disorder, also individuals with relatives with psychiatric disorders, are overrepresented in creative professions such as music and writing. So, but we do not know if this association is causal. You know, creativity leads to psychopathology or psychopathology leads to creativity. And that's what we were interested to find this using a genetically informed study. So previous GWASs of creativity, like there is one GWAS that has been published early this year, I guess, in a very small sample of 405,000 Chinese undergraduates. Apart from that, surprisingly, there are no large-scale GWASs have been conducted today, which is surprising given that this is really a fascinating phenotype. So we thought we decided to do a GWAS for creativity. We studied 3 cohorts, Million Veteran Biobank and UK Biobank, and also the Vanderbilt BioView DNA repository. So how did we define creativity in MVP and UK Biobank? We define creativity based on self-reported creative occupation. So this is the occupation classification. Luckily, it is exactly same in both MVP and UK Biobank. So what we did is we chose this particular category, which is called arts, design, entertainment, sport, and media cases. and the rest of the occupation categories as control. So here I would like to explicitly state that by choosing rest of the occupation categories as control, we do not mean that people in this category are not creative. It's just we expect that in this particular category, we will expect an enrichment in people with creative people. So that's a fair assumption, I guess. So in Vanderbilt, we had a really better phenotype. We went through the patient record, patient case history manually, and then we identified everyone who are professional musicians. The sample size, we had around 3,000 cases in MVP, around 8,000 cases in UK Biobank, and an ample amount of controls in both the biobanks. In Vanderbilt, we actually found 1,800 professional musicians. That's really a lot, thanks to Nashville, America's music city. So this is the overall study design. So we did a GWAS discovery sample, GWAS in MVP and UK Biobank, meta-analyzed it. Then we used the BioView as a validation for polygenic score analysis. And also finally, we meta-analyzed all 3 GWAS and used the resulting summary statistics for a secondary analysis. We studied the genetic architecture in terms of variants, genes, gene sets, and pathways, but we also studied the pleiotropic associations with other traits using analysis such as genetic correlations, Mendelian randomization, etc. So first we did the GWAS in MVP and UK Biobank individually. This did not identify any genome-wide significant hit. We adjusted for age, sex, and also education attainment because these individuals had on average higher education attainment compared to the controls. So though we did not find any genome-wide significant hit, when we looked into the genetic correlation between the 2 cohorts, surprisingly, we found very high genetic correlation. So that was really convincing. It says that we are capturing very similar genetic signals both in MVP and UK Biobank. So we meta-analyzed that, and the meta-analysis also did not find any genome-wide significant hit, but we captured a significant amount of polygenicity for creativity, which we showed through the validation analysis in BioView. So we constructed polygenic score for the individuals in BioView, and we compared the polygenic score for creativity between musicians and controls. And this analysis showed that musicians had a higher amount of polygenic score for creativity compared to controls. And this suggests that what we captured in the MVP and UK Biobank is some polygenic signal for creativity, and it is reproducible. Hence, we went forward to do a meta-analysis of all the 3 cohorts, and this meta-analysis actually resulted in one genome-wide significant hit in chromosome 12. And this is a solid hit. It actually, the index variant at this locus has similar effect sizes across 3 cohorts. And this locus is actually a non-coding locus. We do not know what genes are involved, but interestingly, it seems that deletion of this locus actually leads to, actually is associated with intellectual disability. So it says that this locus has something to do with brain development, and that is really convincing. And we did not find any evidence for population stratification, and the heritability based on LD score regression ranged between 6 to 11% based on different population prevalence assumptions. So I'm going to talk only about the pleiotropic associations with creativity for today due to time constraints. So we did pleiotropic association using LD score regression and Mendelian randomization for almost 65 traits belonging to 6 categories that we think is related to creativity. Of this, I'm going to present only the results for the psychiatric disorders today. So as expected, the bivariate LD score regression showed positive genetic correlation with almost most of the psychiatric disorders. But however, we also found strong genetic correlation with education attainment, 33% correlation. So we wanted to make sure that these correlations are actually unique to creativity and it is not driven by the overlap with education attainment. So to do this, we actually did genomic SEM based analysis where you can do actually a multivariate LD score regression analysis. Let's take schizophrenia as an example. So in a simple bivariate LD score regression, creativity shows positive correlation with schizophrenia. Education attainment also shows a small but significant positive correlation with schizophrenia. And this correlation, positive correlation, is really paradoxical, as we know that education attainment has a strong negative phenotypic correlation with schizophrenia. And we do not know why genetically we see a positive correlation. So the multivariate LD score regression showed that after removing the creativity signal from EA, the education attainment is no longer positively correlated with schizophrenia, but we see a small but significant negative genetic correlation, and the genetic correlation between creativity and schizophrenia became even stronger. So what this means is that the education attainment, the positive correlation between education and schizophrenia was due to that education attainment GWAS actually captures a small portion of the creativity signal too. And this was like really an interesting finding. So similarly, we did this multivariate LD score regression for all the psychiatric disorders, and we see that all the psychiatric disorders shows positive correlation with creativity. And interestingly, even ADHD, which actually we know that has a strong negative correlation with the IQ and education attainment, shows significant positive correlation with creativity. That was really fascinating. The strongest correlation was seen for bipolar disorder, and the least correlation was seen for autism spectrum disorder. So next, we wanted to know if these relationships are causal. So we did a Mendelian randomization analysis. So here in this plot, we plotted the odds ratios from inverse variance weighted analysis. So in the direction from creativity to psychiatric disorder, we did not see any significant associations in any of the psychiatric disorders. But in the direction from psychiatric disorder to creativity, we found a significant association, causal association between psychopathology factor, general psychopathology factor that captures, indexes the genetic susceptibility to multiple psychiatric disorders. So this means that increased risk for genetic risk for general psychopathology factor actually leads to an increase in creativity, but the other way is not true. And this association is consistent across various sensitivity analysis, as you can see here. But a word of caution here, because our creativity GWAS is not really that strong GWAS. So probably in future, when we have a better phenotype of creativity and a strong summary statistics, this picture might change. To summarize, using a shallow phenotype, we partially capture the polygenic signal for creativity. The heritability is extremely low compared to twin-based estimates. So possible reasons we think is because of the superficial nature of the phenotype, and also the socio-cultural influences that we haven't accounted for in the analysis. The polygenic signal that we captured, though minimal, consistently replicated across 3 cohorts. That's really convincing. And the pleiotrophy association showed, as expected, a positive genetic relationship with almost all the psychiatric disorders. And importantly, this is independent of education attainment. And Mendelian randomization analysis suggests that psychopathology leads to creativity, but not other way around. Thank you. So I'd like to acknowledge my collaborators, and I have to specifically state that every collaboration in this project happened in the networking session in World Congress in last year. And I specifically like to thank Panos Roussos, who is the main supervisor of this project, and he kindly let me to take the lead for this project. Thank you.
Thank you. Very interesting presentation, Veera. So there are any questions, please put them in the Q&A feature. There's one, maybe you can answer that about how can it differentiate creativity from other possible traits associated with profession like art? Shouldn't you?
So I'm not sure if there is a difference between this. So I guess the creativity is something that is common to all the professions of art, like music and painting. Probably there might be differences, but I'm not sure. So we don't have data to, probe those differences. for now, the assumption is that creativity is something that is common across all the art fields, painting, music, writing, acting, etc.
Yeah, so maybe a quick follow-up question for me. Did you ever think about having like an alternative profession as control, like some bureaucracy or some more boring type of profession? More, yeah. Would that be possible with your list of phenotypes?
Yeah, so for now, we did not do any post-hoc analysis by choosing different controls because that will result in a lot of analysis. And so for now, we are sticking with the simple definition, but definitely, like one of the reasons for the low heritability, we think is because we have included many professions, particularly, life sciences profession, which has more creative people as much we see in our the autistic profession. That might have pulled down the heritability, but that is something we would like to explore as a secondary analysis later.
Yeah, thank you. So I think, Alessandro, due to the time, we need to move on. So maybe, Veera, you could see if you could answer some of these questions about power and overlap with personality in the Q&A answer feature.
Yes,
then we move on. Thanks for questions and your presentation. Alessandro?
Okay, so now we are inviting to talk Chia-Yen Chen from Biogen with a large-scale exome sequencing identify novel genes for cognitive function.
So hi, everyone. My name is Chia-Yen Chen, and I would like to thank the organizers for giving me this opportunity to present our recent work on leveraging large-scale exome sequencing data to identify novel genes for cognitive function. And as the financial disclosure, I am an employee of Biogen. So cognitive function has long been considered as an endophenotype for psychiatric disorders. And what are endophenotypes? Endophenotypes are markers that are genetically correlated with disease liability and can be measured in all individuals regardless the individual is affected by the disease or not. And finally, to the key of genetic discovery, since the endophenotype is an intermediate between the genes and the psychiatric disorders, it can potentially provide better power to identify disease-relevant genes. So in our previous study, we performed a meta-analysis of the two largest published GWAS for general cognitive function using MTAG. And we identified 241 independent loci associated with cognitive function, with 29 novel loci that were not previously identified. And it is worth noting that for these 241 cognitive function loci, they were identified through common variant association in GWAS. And therefore, this study really didn't provide much insight into the potential impact of rare variants on cognitive function. On the other hand, a previous study led by Andrea Gana showed that rare and ultra-rare protein truncating variant burden, PDV burden, in PDV intolerant genes has strong impact on educational attainment in a combined general population sample from Sweden and Finland, as shown here in the left panel. And in addition, they also showed that the PDV burden has a strong impact on intellectual disability early in life and is associated with various psychiatric disorders such as autism and ADHD. So here in our study, we aim to further examine the impact of rare variants and try to identify novel genes for cognitive function by leveraging large-scale exome sequencing data in the UK Biobank. The exome sequencing data were generated by the Life Science Genetics Consortium, which includes 8 pharmaceutical companies, including Biogen. And the exome sequencing data was generated at Regeneron Genetic Center and was processed using Regeneron's SBP standard pipeline. And of note, the first 50,000 exome data were released early last year in July 2019. And you can see more details about this data release through the link over here at the lower right corner. And our current analysis are based on an interim data freeze of 300,000 samples with 23 million QC rare and common variants from the exome sequencing data. And for the phenotype part, we extracted several cognitive function phenotypes and performed detailed phenotyping using these covariates. These phenotypes include educational attainment, reaction time, pairs matching, and verbal numerical reasoning, or commonly known as the fluid intelligence covariate in UK Biobank. And the phenotypes circled in green here, they were collected in all 500,000 UK Biobank participants. And the phenotypes circled in yellow here were only collected in a subset of 200,000 UK Biobank participants. So this difference determined largely the sample size we have for the gene discovery for these phenotypes. And in addition to these original cognitive function variables, we also derived the general cognitive function gene using principal component analysis based on educational attainment, reaction time, and pairs matching. And for gene discovery, in this analysis, we focused on the impact of protein truncating variants, PTVs. So these are genetic variants predicted to shorten the coding sequence of genes. For data QC, we included only PTVs with minor allele frequency less than 1% and filtered out genes with PTV count less than 10. The association tests were done between cognitive function phenotypes and PTV burden for each gene using linear model in unrelated samples of European ancestry in the UK Biobank. And the PTV burden was calculated as illustrated here in the right panel. We sum over the count of PTVs for each gene for each individual. And for the association test, we use the exome-wide Bonferroni corrected significant level to identify novel genes for cognitive function. And so here are some results that highlight from our analysis. So the first one is the genome-wide PTV burden analysis for educational attainment. We identified 2 genes that are associated with educational attainment. And in particular, GIGYF1 has been associated with schizophrenia through GWAS using common variant association, and also autism through de novo mutation. And it is well known that both schizophrenia and autism have some cognitive impairment as a common clinical feature for these 2 disorders. And interestingly, in a paper published last week, led by the plenary speaker yesterday, Matt Hurls, GIGYF1 was also identified as a novel gene for developmental disorders. So put together with our result, it suggests that PTVs in GIGYF1 have an impact on developmental disorders early in life, and also on cognitive function in adulthood in general population. It also suggests a potential lower penetrance for PTVs in this gene. To further investigate the contribution of common and rare variants on educational attainment, we performed polygenic score analysis and genome-wide PTV burden analysis. And as expected, higher polygenic score for cognitive function is associated with higher educational attainment, shown here in the left panel. And higher genome-wide PTV burden is associated with lower educational attainment, as shown here in the right panel. And to really drive the point, we further show that the impact of cognitive function polygenic score and genome-wide PDV burden on education attainment appears to be independent. It is highlighted here by the two lines in parallel, which represents the impact of cognitive function polygenic score on education attainment in high and low PDV burden group. And this is also supported by a non-significant result in a formal interaction test. In our second set analysis, we did a gene-based PD burden association for reaction time. And we identified one gene, KDM5B, that is associated with reaction time. And it is also implicated in intellectual disability and autism previously. And interestingly, we performed a phenome-wide PDV burden analysis test against PDV burden in KDM5B against over 4,000 phenotypes. And we showed that KDM5B PDV burden is also associated with multiple muscle-related phenotypes, such as hand grip strength, IGF-1, which is related to the growth of bone and muscle, and also lung function. And this result highlights the potential pleiotropic effect of PDVs in KDM5B. And finally, in a smaller subset sample, as we mentioned earlier, the total sample size here for the verbal numerical reasoning is around 100,000 individuals. And in this analysis, we identified two novel genes for verbal numerical reasoning. And I would like to highlight that ANKRD12 was associated with both educational attainment and verbal numerical reasoning in our analysis, which suggests a consistent association signal with two analyses using complementary phenotypes. And also, the gene CAPRIN1 was implicated previously in neurodevelopmental disorders, which is consistent with our findings here. So in summary, we identified 4 novel genes influencing cognitive function in adult general population. And among these 4 genes, GIGYF1, KDM5B, and CAPRIN1, these 3 genes have been previously associated with autism and other developmental disorders. Our analysis adds to the association between these 3 genes and cognitive function in adult population, which suggests a continuum of phenotypic presentations of PDVs in these genes and potentially lower penetrance for PDVs in these genes. And our phenome-wide analysis showed an interesting result of pleiotropic effect of PDVs in KDM5B, which affects both cognitive function and muscle-related phenotypes. And our polygenic score and rare variant PDV burden analysis showed independent effects on educational attainment. And finally, I would like to highlight that our analysis showed the value of large-scale sequencing in well-phenotyped cohort for gene discovery beyond GWAS, such as UK Biobank. And in our next step, we will expand the analysis to the full UK Biobank sample for better power for gene discovery. And here I would like to thank the participants of UK Biobank. And this project is really a joint effort of my colleagues in translational biology at Biogen, and Max Lam and Todd Lenz at Zucker Hillside Hospital, and Tian Ge and Hailiang Huang at Mass General Hospital and Broad Institute. Thank you.
Thank you, Chia-Yen. Very, very, very interesting. And hopefully when the UK Biobank whole sample will have exome sequencing would be much more interesting. We have one question that was also my question, honestly. So Liu Dongling is asking the question about variants. And in fact, I also was wondering why focusing. I think it's important, of course, protein truncating variants is important. It's very conservative, of course, because it's a severe damage. But me and the panelists, we are wondering, what about the other variants? Are you considering to consider also less damaging rare variants? Some scores?
Yes, definitely. So this is actually the first step of our analysis. So for full disclosure, we now do have close to the full UK Biobank sample exome sequencing data, and we do intend to expand analysis beyond PDV to missense burden, damaging missense burden, and benign missense burden for these phenotypes.
Yes, that would be challenging because it's difficult to give a priority score. So just the presence or absence of these variants, or if they have different weights compared to the most severe ones.
Yes, I'll probably refer to the schema paper where we can classify the missense burdens using CAT or MPC or such kind of prioritization scores and distinguish out the damaging and damaging and benign missense variants.
Exactly.
That's
wonderful.
Yeah.
So I think we are going to the next speaker. So I give the word again to the co-chair, Roland Razen.
Thank you. We are moving on to another endophenotype. Now it's suicide attempts, and the speaker is Dr. Jooeun or Jooeun Kang from Vanderbilt University, and the topic is unraveling the genetic architecture of suicide attempts and psychiatric disorders. Please.
Thank you for the introduction, and thank you for the organizers for allowing me to speak today. The subject of my study is suicide, and it is a public health problem. In the US, it's the second leading cause of death among individuals 10 to about 34, and 2017 alone, there were 47,000 plus deaths. And in the United States, it's an especially dire problem because the rates of suicide has been increasing for the past decade, and this trend has been seen nationwide. And as geneticists, we are interested in studying this because it is heritable. And so the upper limit of heritability for suicide attempt has been estimated to be about 55%. And the lower limit of heritability from GWAS, from common variants, has been estimated to be about 5%. Now, when we talk about suicide attempt, it is important to consider the complex relationship between suicide attempt and psychiatric disease. And this is emphasized by the fact that about 90% of people who die from suicide, those deaths occur in the presence of a mental disorder. And to illustrate the complexity, let's focus on major depressive disorder, which is the most common psychiatric diagnosis among individuals who attempt suicide. For MDD, about 1 in 5 individuals with MDD will attempt suicide in their lifetime. So when we think about using genetics in order to distinguish and predict individuals who attempt suicide, it's important to consider what are the genetic risk factors that sets this individual with MDD apart from the others who did not attempt suicide. So one hypothesis is that suicide, there is shared genetics between major depressive disorder and suicide attempt, and that suicide attempt is its own entity that has a lot of pleiotropy with MDD. And the other hypothesis is that suicide attempt is an extension of MDD and is an outcome of severe MDD. So to answer this question, prior studies have looked at heritability estimates for suicide attempt, adjusted for psychiatric diagnoses, and observed what happened to those heritability estimates after adjusting. And what they saw was a decrease in heritability, and we can learn 2 things from here. The decrease itself shows that the genetic etiology of suicide attempt is partially shared with psychiatric diagnoses. However, the remaining heritability shows that there is a partially distinct genetic architecture of suicide attempt as well. And so in order to elucidate further this genetic architecture of suicide, the International Suicide Genetic Consortium was formed. It consists of 15 different cohorts from 4 different continents. It consists of about 30,000 samples from 3 different ancestries. So when we're doing our meta-analysis of suicide attempt, we're pulling cases from 2 different sources. So we're pulling suicide cases from large biobanks. So in this case, our suicide attempt cases are agnostic to whether this individual has psychiatric diagnosis or not. However, we're also pulling cases from psychiatric diagnoses GWAS. So every suicidal individual here, has a psychiatric diagnosis, and those include MDD, bipolar disorder, schizophrenia, and anorexia nervosa. And everyone who has not attempted suicide or controls, and the sample size are shown in the slide here. Now with that primary suicide attempt GWAS analysis, we can ask the scientific question of how shared is the genetics of MDD with suicide attempt. And the reason we're focusing on MDD in particular is because the prevalence of MDD among individuals who attempt suicide is very high. And also, we know that there is polygenic overlap between suicide attempt and MDD, and this has been shown with significant genetic correlation as well as polygenic risk score associations. So the way that we did that was we applied MT-COJO, and what that does, it's a statistical method that takes the effect size from the suicide attempt GWAS. It conditions those effect size for MDD's effect on suicide attempt, and thus it results in a suicide attempt condition on MDD effect sizes. Taking that one step further, we wanted to look at the suicide attempt specific genetic architecture by study design. And so we did that by pulling our suicide attempt cases from just the psychiatric diagnosis GWAS and individuals who have the same psychiatric diagnosis but who have not attempted suicide or deemed as controls. And the sample size is shown here. Diving right into the results, so this is a Manhattan plot of our primary suicide attempt GWAS analysis. It yielded 2 genome-wide significant loci, one in the MHC region and one in the introgenic region in chromosome 7. And our heritability estimate from this GWAS was about 7%. When we conditioned for MDD, we saw a decrease in SNP heritability from 7 to about 4.5%. That decrease is anticipated from given the prior studies that we've seen before. And then also the genome-wide significant loci went from 2 to 1, and that 1 was the introgenic chromosome 7 one. The suicide attempt within diagnosis GWAS was underpowered to detect any genome-wide significant loci. However, when we looked at the heritability estimate, it was similar to the condition on MDD model, and also the genetic correlation between the two was very high and significant. And what this told us was that this validated that the suicide attempt condition MDD was able to sufficiently capture the suicide attempt specific genetic architecture. And we reached the same conclusion by doing a PRS analysis where the suicide attempt condition in MDD PRS was able to effectively capture suicide attempt among individuals with psychiatric diagnoses. Now, if you remember, there was that one genome-wide significant loci in chromosome 7. So we looked at the effect size of that loci in suicide attempt and in known suicide attempt risk factors. And what we see here is that with our primary GWAS analysis and after conditioning, this loci has the largest effect in suicide. And even among those in other non-psychiatric traits, it's much lower. And the significant p-values are explained by pleiotropy. And this loci also has much lower and less significant effect sizes in other psychiatric disorders. Now going back to that question of whether suicide attempt is an extension of MDD or whether it's a separate entity that has a lot of pleiotropy with MDD, we try to look at this question by comparing side by side the genetic correlation of suicide attempt with psychiatric disorders before and after conditioning. And so if the genetic correlations do not change after conditioning, that means that that genetic correlation is not driven by MDD. Whereas if there is a drop in genetic correlation, that correlation is driven by MDD. So what we see here is that for the most psychiatric disorders, that there is significant genetic correlations with suicide attempt to begin with, and that for the most part, those genetic correlations are not driven by MDD because they're the suicide attempt, the orange and the purple are pretty similar. But there are also ones that do change after conditioning. And so one, that's MDD. So that drop is explained by the statistical method itself, since we're conditioning out for the effect of MDD. And the remaining genetic correlation after conditioning just shows how much pleiotropy there is with suicide attempt genetic architecture and MDD. Also of note is that there's a drop with autism spectrum disorder, and also with self-harm ideation, it seems that genetic correlation is also somewhat driven by MDD, but not to a significant extent. Now we did the same analysis, but we expanded it to genetic correlations with suicide attempt risk factor traits. So these include substance use like alcohol and smoking, education levels and socioeconomic status, and also pain and psychiatric traits. And we see that for the most part, these genetic correlations remain the same as they're on, this is a correlation plot. But there's also this group of traits that do change, a decrease in genetic correlation after we condition for MDD, and that group mostly consists of psychiatric traits. We're able to reach the same conclusion when we look at the top genetic correlations with suicide attempt and see what happens to them after we condition an MDD and rank them by how much they change. So what we see here is a distinct group of depressive traits, or traits that are related to depression, that decrease the most when we condition an MDD. And that makes sense. This makes in that these genetic correlations with suicide attempt are driven by MDD. What was also notable is that there are these traits that either do not decrease as much or don't decrease at all. And these consist of traits that are related to socioeconomic status. So these include health satisfaction, financial difficulties, and also notably the Townsend Deprivation Index. And also traits that are related to smoking. And so what this tells us is that the genetic correlation with smoking, socioeconomic status, and suicide attempt are not primarily driven by genetics of MDD. So overall, our consortium was able to conduct the largest meta-analysis GWAS of suicide attempt to date with about 30,000 cases. It yielded us 2 genomically significant loci and a significant heritability estimate. And by conditioning on this GWAS on the genetics of MDD, we're able to look at the suicide attempt specific SNP effects. And when we did, we saw that most genetic correlations of suicide attempt and psychiatric disorders are actually not driven by MDD, but it's also notable to know that after conditioning, there was still high genetic correlation with MDD, which just shows how much pleiotropy there is between suicide attempt and MDD. We also saw that there is positive genetic correlations with measures of socioeconomic status and smoking, and these did not change after conditioning, which suggests that there is shared biology between suicide attempt and these risk factors that are not mediated by MDD. So in terms of future directions, we would like to examine the genetic architecture of the whole suicidal behavior spectrum, so ideation, attempt, and death. And this is important to consider these separately because while they are a spectrum, one does not necessarily lead to the other. So it will be important to consider them separately. So I'd like to thank our collaborator, Neve, my PI, Doug, and everybody listed on here who gave us their important samples that made this consortium happen. And thank you very much.
Thank you, Jay. Very interesting presentation. So we are running out of time as far as I see, but maybe one quick question. So
I'm not seeing any questions on the Q&A box.
No. So I have one. So how with these small sample sizes, have you looked into some other phenotypes? There, I think there is also increased suicide for in general in mental illness. Have you tried to combine samples, like with the big cell paper, the combined analysis across everything.
So I think you're talking about the cross disorders,
Yes.
Wes. So I think the thing is, I think different disorders have different overlap of genetic architecture with suicide attempt. So we actually looked at the correlations with the cross disorders, GWAS, but the thing is, I think the way that the effect size for the cross disorders analysis was performed is not necessarily a meta-analysis per se. And so I think the method in which the effect size, it wasn't kind of I don't think that was a proper way of looking at just the general psychiatric diagnosis contribution. So we instead opted for the more diagnosis specific ones. And also there are different prevalences of suicide among different disorders. So I think that also we thought was another reason for us to look at disorders specifically rather than just look at the broad stroke psychiatric diagnoses.
Thank you. Alessandro, you take over.
Yes, so thank you, Jay, for your nice presentation. It's very important to focus on suicide, so difficult to study. And I think we are out of time. So wonderful, wonderful speakers. I remind all the attendees that the registration, the recording is there. From tomorrow, you can see for a couple of months. And if you want to ask further questions to the speakers, you can meet them and privately discuss. I think they're really available. So thank you very much for the attendance and enjoy the rest of the meeting and the plenary lecture that will be later. Andrea Meyer-Kindenberg would be very interesting, I think.